very sweet, very thoughtful, and very special. And it's very special for me to be able to have everybody here today to talk about what I've been doing for the past few years with my thesis work. So I'll jump in to talking about inhibition and state-dependent cortical function. So what I'm going to be telling you about are two separate projects that are both out now as preprints and under or in revisions right now in different journals. I'm going to go really in depth into one of the projects that I worked on during my PhD, but also for sake of completeness here, I'll also give you a brief flavor of the second project. And as Renata said, I was one of the first PhD students to join the lab alongside Claire. And being the first PhD student, I got to interact with a lot of the different pillars that make up the P.h.D. lab. Some of these are the regulation of behavioral states, the diversity of cortical inhibitory neurons, as well as the development of the cortex in early postnatal periods. And these projects that I worked on touched in all of these different areas, which I'll move through here and tell you a little bit more about. The first, regulation of behavioral states, as you might be able to guess by this picture, some of the most dramatic examples of different behavioral states are the differences between sleep and wakefulness. This has really profound impacts on the behavior of an animal, as well as brain function. Behavioral states vary in many different dimensions besides just sleep and wake. There's periods of active locomotion, as well as periods of intense alertness, such as if you're up here trying to find your external committee member right before the presentation. But we focus on this, on how behavioral state interacts with vision. And in sort of a classical vision experiment, one that we do in the lab using a mouse, we might show a mouse several different visual stimuli, a drifting grating, maybe some cheese, or a little cartoon of a cat. And classically, people have looked at what's called the tuning, or how different cells will respond to these different visual stimuli. And you might see something like this, where there's certain tuning to this drifting grating, different tuning for the cheese, and then maybe it doesn't really like the cat very much. And these, for many years, were thought to be fixed, regardless of when you showed an animal a specific stimulus. The brain's always going to respond in the similar way. But more recently, in maybe the past 10 years, it's become appreciated that in different behavioral states, these responses are going to be slightly different. The tuning will be the same. If the animal is moving around, you'll still like these two and maybe dislike this one, but the gain is going to be different. The magnitude of these responses will be different. And this was a big change, and one that's rather recent in our understanding. And our lab really is interested in understanding more about the underlying circuitry that produces these different changes. And in order to do that, we focus a lot on this inhibitory diversity, which I mentioned. We've known for a really long time that the different cells in the brain come in many different shapes and sizes. Work from nearly 100 years ago from Santiago Ramon y Cajal, which he won a Nobel Prize for, showed us with beautiful hand drawings of individual neurons in the brain that they come in a variety of different shapes and sizes, and were hypothesized for a long time to have different functions, but these functions remain somewhat inaccessible for many years. As I said, we're interested in understanding the visual system. We use the mouse as our model for it, and we also want to understand this during periods of development. And the mouse is an interesting system for this because when mice are born, they don't actually see.
How's it going?
Hey, Emily, I think you're unmuted.
Oh, sorry.
It's doing well, Emily, but
So when mice are born, they can't actually see, but there's a lot of interesting processes that are happening that are preparing the the brain to first experience this visual information. This period in mice corresponds to about the second trimester in humans, and there are shared features. One of those important shared features are these periods of spontaneous activity where the retina is active in the absence of very much light input. And these waves of activity travel across what is the surface of the retina and propagates this activity throughout the visual system, shaping many, many different developmental processes. And this is what one of the projects that I worked on in the lab was focused on, understanding these retinal waves and how it interacts with multiple levels of development within the visual cortex. We were interested in gene expression, we were interested in function, and we surveyed a lot of these different levels. We used a mouse model that reduces these retinal waves, and unlike other retinal wave knockout models that are used in the field, doesn't have some of the compensatory activity that can be found. We surveyed gene expression and found that across the diversity of different cell types in the cortex, gene expression was highly disrupted across many cell types. We looked at synaptic connectivity, which was disrupted. We looked at function, and particularly with state-dependent function, and found that in particular cell types, in this case layer 1 inhibitory neurons, state-dependent function was disrupted. And then finally, looking in pyramidal cells, we found that this behavioral state-dependent gain that I told you about was also disrupted in this mouse model. And so we showed that these broad changes that we observe all sort of coalesce into finding this state-dependent disruptions, for the first time linking spontaneous activity during early development with this phenomenon. So this will be all that I tell you about for this first project. You know, if there are any questions, you can ask me now. You can ask me later, and I can tell you more. But I'd really like to give you a flavor, an in-depth flavor for the major thrust of my PhD work. This work focuses more in on this diversity of inhibitory neurons that I told you about. For many years, since the 1900s even, this morphological characterization and classification of inhibitory neurons was nearly the gold standard for defining similar groups of cells into cell types. But about maybe 2 decades ago, there was a really dramatic shift where we started using genomic information, looking at the genes and markers that different cells express to start to group them into different categories. A really big change happened with the application of a particular technology called single-cell RNA sequencing to brain tissue. We were able to read out all the different genes that were expressed in individual neurons to be able to group them into different class categories. We, at the end of this work, or during this work, the people who were doing it were able to come up with taxonomies that looked like this. Don't worry about this big complicated slide. Most of it doesn't matter, but I'll walk you through it. They were able to group all the inhibitory neurons into the brain into these discrete bubbles, which are distinct cell types, and really show that there was a lot more diversity than maybe people had been thinking at the time. But a lot of these groups or a lot of these different cell types that they were classifying were somewhat unknown. It wasn't really clear what exactly the function of these distinct groups was. When this came out, a lot of groups, including ours, was interested in beginning to tackle this heterogeneity and to figure out what the functional role of these distinct groups were. Finding the entry point into this is maybe a little difficult. There's a lot of different types of cells that you could study here, but we chose the rationale of looking at the really strange distinct cell types in these taxonomies. And you can find a few of these in this group. One of them is this little island here, these PV-VIPR2 cells, which some of you might know, or the classic chandelier cells, which have this really beautiful and distinct morphology and provide somewhat of a grounding for this genetic-based classification. But the ones that we focus on are these grouped down here. These cells express two genes that mark them, somatostatin and chondrolectin. I'll call them somatostatin chodal cells for this talk. For the experts, these cells also express NOS1, they express TACR1, they express muscarinic acetylcholine receptor type 2. I might call them NOS1 or nNOS cells at some point. I hope you forgive me if I slip up. So yeah, we really wanted to understand this group. I'm showing you another representation of this data from the last slide. Here, where each dot is one cell that's gone through the single cell sequencing pipeline, and these dots are placed on this map next to one another based on their similarity of gene expression. So this island up here are the somatostatin chodal cells. They're all clustered tightly on top of one another, and they're clustered away from the rest of these other inhibitory neurons, suggesting that their gene expression is really quite distinct from other inhibitory neurons. And unsurprisingly, they have other really unique features about them. These cells, unlike other locally projecting interneurons, have long-range projections that travel long distances across the brain, really setting them apart from other inhibitory neurons. And given that this population is so small, less than 1% of all the inhibitory neurons in the cortex, evolution over the years might have eliminated these if they weren't doing something important. But in fact, the opposite is true. These cells have been around for quite a while. They can be found in species as diverse as newts, amphibians, reptiles, mice, primates, and humans, conserved across 350 million evolutionary years, suggesting that they've been kept around to do something important. So we want to understand what that important thing is, and we want to answer a few questions to better know these cells during my PhD work. We wanted to answer the where of these cells, where are their downstream targets, and what brain regions are they able to affect. The when of these cells, when are they active, what are their patterns of activity. And finally, the what of the cells. What is the functional role? What is it that they're doing in the brain that for which evolution is keeping them around? And in order to study this, we use our good friend the mouse. We use him for a few reasons. These cells were largely understudied when we first began this project. And that was because there weren't really very good tools to be able to target and manipulate and better understand these cells. But using mice, and in particular using a new set of tools at the time, intersectional genetics, we were able to target these cells based on the co-expression of two different genes, somatostatin and NOS1, which is coming up here again, and targeting the cells at the intersection of the expression of these two genes, which allows us to target and manipulate our somatostatin chodal cells. So the first thing we were trying to answer was this where question of the cells. Where do they project to? What are their downstream targets? And to do this, we used a tool that allowed us to fluorescently label these cells, as well as their processes throughout the whole brain. This was one of the first images that we got from one of these experiments, where we labeled just one somatostatin chodal cell in this thin section through the brain. And you can see, even though we have this one cell, there are these really massive arborizations that are going all over the place around this cell, suggesting that they have the potential to influence a lot of downstream circuits. We wanted to expand upon this experiment and did a similar experiment. I hope this is coming up all right. But here we've done the same thing. We've injected this tool, which allows us to label a variety of these somatostatin chodal cells around an injection site. We can see those same dense projections in this clarified mouse brain, the local projections around the injection site, as well as these long-range projections that travel to a variety of different areas all across the brain. We wanted to quantify these images, and we did so with the help of a really talented team of undergraduates and local high school students who really put in a lot of work to take these images where we have our labeled cells, reconstruct all of the arbor that we can see in these cells. We register this reconstructed data to atlases of the mouse brain, allowing us to know what areas these projections are in. We quantify these projections. One of the first things that we did was we looked at the layers that these projections were found in. The local projections around the injection site were spread pretty evenly across all layers, whereas these long-range projections that are traveling across the brain really seem to like to travel in the upper layers, particularly right along the surface of the brain. We also looked at the areas in which these cells project to, and when we labeled these cells in primary visual cortex, the arbor seems to like to stay within visual cortices and higher visual areas, but this arbor also has massive projections elsewhere in the brain. It targets pretty much every neocortical area in the brain on the same side as the injection, minus the most frontal and most dorsal areas, with a small projection over to contralateral visual cortex. As I said, these projections really like to stay within the neocortex. And in all of the experiments we did, we never found any projections outside of the neocortex, minus a small but significant consistent projection to the subiculum. Here we were looking at these sort of bulk projections, but we also wanted to, from the very beginning, we wanted to know if there was any logic for single cells and how they project to these different areas. During my PhD, we never got to answer this question, but during this revision process, we've teamed up with the Allen Institute, who were able to do these very detailed single cell reconstructions of the somatostatin choroidal cells. We're collaborating to learn the patterns by which single somatostatin choroidal cells will choose their downstream targets. So this together gives us a really detailed picture of where the projections are going from these cells. We also want to know what cells what downstream target cells, target neurons, these somatostatin choroidal cells interact with. To answer this question, we teamed up with Stephanie Rudolph and Stefano Luzzo in her lab. He looked at which downstream cells are targeted by the somatostatin choroidal cells by expressing a channel rhodopsin molecule that allows us to activate the axons of the somatostatin choroidal cells while recording from potential downstream partners, which are shown here in black. We were really surprised when we started doing these experiments. Pretty much every cell that Stefano patched onto to record from seemed to be connected to the somatostatin choroidal cells. All the cells in layer 6, layer 5, layer 1, and most of the cells in layer 2, 3 received input from these cells. And there was only the small population in layer 2, 3 that was not connected. These inputs are fairly consistent in size across most layers, and across inhibitory neurons as well as pyramidal cells, but layer 6 appears to receive fairly strong input from these cells. Altogether, this is painting a picture of these cells that have these really broad projections targeting many downstream neurons, and therefore able to have a really strong impact on many cells within these cortical networks. So the next question we turn to after figuring this out is trying to ask the when of these cells. When can they influence these large downstream populations? And when we began, there was not much known about these cells, but there definitely were some guideposts that were crucial for beginning to understand these somatostatin choroidal cells and when they might be active. I'm showing you here some work from Tom Kilduff's lab, who I believe is on Zoom right now. He published some works showing that these cells are likely to be active during periods of intense slow-wave sleep. When they sacrificed rats, I believe in this case, that had undergone either intense periods of wake, long-term sleep depression, or had recently undergone intense periods of sleep, recovery sleep after the sleep deprivation, this histological marker of activity, FOS, strongly colocalized with somatostatin chodal cells and only in these sleeping animals, suggesting that this intense periods of slow wave sleep are when these cells are active. But this experiment leaves something to be desired. As I told you earlier, these behavioral states vary dynamically on a minute-by-minute basis throughout the life of an animal. And this experiment is just giving us one time point for that entire animal. We wanted to understand on this moment-by-moment basis how these cells were turning on and off. And we wanted to divide our states in a bit more fine-grained way. One of the ways that we do this in the lab is using pupillometry. Pupillometry is a good reflector of the behavioral state of the animal. When an animal is more quiet, sitting stilly, pupil is largely constricted. And as the animal starts to become more alert and aroused and moving around, pupil dilates. If you're recording cortical activity at the same time, you can see that there are big changes in the activity of the cortex that co-occur with these changes in state. During these quiet periods, the cortex is largely synchronized or dominated by low-frequency rhythms in the local field potential, electrical activity of the brain, and spiking activity. And as the animal starts to become more alert, these cortical networks become more desynchronized or dominated by high-frequency activity. We took an approach where we really broke apart these different behavioral states into very discrete chunks. We divide between wake and sleep, between active moving periods, which in mice correspond to whisker twitching, to quiet waking states where the animal is sitting still and is awake, not quite asleep, which are somewhat synchronized. They have these low frequency rhythms present. We parse out slow wave sleep or non-REM sleep, which is when some of the most deep synchronization occurs. And then finally, we can parse out REM sleep, where most dreaming happens in humans, which is a sleep state, but is not a synchronized state. So we wanted to observe the activity of somatostatin chodal cells across all of these different states. And to do so, we needed a preparation that would allow us to do this. We turned to a technique called 2-photon calcium imaging to be able to record the activity of this really, really sparse population of cells within the brain. And we also wanted these animals to be awake and sleeping at the same time. So we came up with this, or I set up our 2-photon microscope with all these crazy apparatuses to be able to head fix a mouse underneath our 2-photon microscope objective while being able to record local field potentials, electrical activity in the brain, monitor its locomotion, and do videography to see its face and pupil activity. With all this, we still wanted our animals to sleep, and it took a little bit of fussing, but we finally came up with a preparation. Here's one of the first animals from this preparation, where you can see that the animal will fall asleep, it stops moving, enters a period of deep synchronization, even enters REM sleep briefly, and then wakes back up and starts moving around. When we started to combine this with recording of these somatostatin chodal cells, we started to see data coming in that looks like this, where we were monitoring pupil simultaneously with measuring the activity of a somatostatin chodal cell. And when the pupil was really dilated, the animal was alert, the somatostatin chodal cell was largely off. But as the pupil starts to constrict and the animal starts to relax a little bit, the somatostatin chodal cell starts lighting up. Its activity is telling us this lighting up is telling us that it's active when the animal is less aroused, less alert. We measured this many times over, adding a number of different measures: local field potential, EMG for muscle activity, facial movement, locomotion, pupil, while recording many of these somatostatin chodal cells. We get data that looks something like this, where we're able to break it apart into our different states: non-REM, quiet wake, movement, REM sleep. And when you look at these defined periods, say when the animal is in a low arousal state, like non-REM or quiet wake, these somatostatin chodal cells are really strongly active. They're pretty much always on. And their activity seems to really match the synchronization state. during these periods. It correlates very strongly with delta amplitude, a measure of synchrony. When the animal is in a less synchronized state or it's moving around, it's more alert, these cells are really strongly suppressed. There's almost no activity in the somatostatin chortle cells during this period. And then finally, looking at REM sleep, these cells are also largely inactive. This sleep state, this non-synchronized sleep state, the somatostatin chortle cells are really suppressed. Looking across many cells, this pattern is quite consistent. Cells are most active during periods of non-REM sleep, followed by quiet wakefulness, and then strongly suppressed during periods of movement and REM. The activity of these cells correlates with our measurements of state, as we might expect. It really strongly correlates with the level of synchrony in the cortex, and negatively correlates with these measurements of high arousal, suggesting that these cells are active specifically during these low arousal synchronized periods. We wanted to know how unique this was to our somatostatin chortle cells. We're picking out this really fine-grained population. We want to know that this is something, what we've observed so far is something unique to these cells and not something that's a general feature. So we looked in an open data set from the Allen Brain Observatory where they had imaged the somatostatin population of inhibitory neurons as a whole, while measuring some of the similar measurements of state that we do, locomotion and pupil. And as we expected, the somatostatin population as a whole, which has many more cells than the somatostatin chortle subgroup, they are positively correlated with arousal state. They're active when the mouse is alert and moving around. Their activity positively correlates with locomotion as well as pupil area. The majority of the cells that we observe are positively correlated, but there's also a small population of cells that are consistently negatively correlated to locomotion and pupil area. This, very interestingly, the proportion of these arousal anti-correlated cells is almost the exact same proportion that the somatostatin chortle cells are within the somatostatin population as a whole, suggesting that maybe we're getting these somatostatin chortle cells in this experiment. This is really showing that our somatostatin chortle cells are quite unique in the way that they're active and inactive. They're quite unlike the other somatostatin neurons in the brain. And knowing this, we then turn to our last question: the what of these cells? What exactly is it that they're doing? And given that they're so strongly correlated with the synchronization state, we thought to ask whether they're potentially causal in generating these synchronization state. Maybe the activity of these cells is the thing that's synchronizing cortical networks, or at least promoting it in some way. But this ran into somewhat of an issue with the literature of how sleep is generated. So sleep, a really synchronized state, has many papers that have been written on the circuits that are involved with the generation. Many reviews, many images that are similar to this one that I picked from this review from Weber and Don in 2016, where they show all these interacting nodes in different circuits that are supposed to generate the different states of sleep and wake. And one of the really consistent features that you see in these reviews and in some of the thinking of the field is that these sleep and wake states are generated primarily by subcortical circuits. It's the interaction of these subcortical nodes that are generating some sort of neuromodulatory milieu that determines whether you're asleep or awake, and the cortex is just following along to what these subcortical circuits are doing. We think this leaves somewhat of a hole. We think it maybe leaves a somatostatin chortle-sized hole up here for the regulation of these synchronized states, which I'm going to zoom into here. As I said, these synchronized states are dominated by periods of low frequency activity, where the electrical fluctuations occur in the cortex in this frequency range. And the spiking activity, the activity of individual cells, follows along with these low frequency fluctuations. And we think the somatostatin chortle cells are really going to play an important role in the dynamic regulation of these synchronized states, because it's not just one monotonic state. Slow wave sleep or synchronized states are not just one thing. they vary based on many features. They vary based on the amount of sleep you've gotten the last night. They vary across space in the brain as well as with the amount of time that you've spent in a particular state. And they vary according to usage. There's some classic work out there showing that if you're doing a task where you're using maybe one arm very, very intensely during the day, when you sleep the next night, the brain area that's controlling this one arm is going to sleep more intensely. It's going to be more intensely synchronized during periods of slow wave sleep, suggesting that there's some local mechanism for regulating synchrony, such as the somatostatin chortle cells. So we wanted to test this. And to do that, we turned back to our intersectional genetic tools. We expressed a channel rhodopsin in the somatostatin chortle cells, which allows us to turn these cells on by shining blue light onto the brain. So we recorded brain activity using these linear silicon probes, which have 64 channels that span across all the layers of the mouse cortex, allowing us to record local field potentials as well as spiking activity across all the layers that we're able to stimulate with these tapered optical fibers that are also inserted into the brain. We looked at many different measures of the synchronization state of the cortex during these experiments while light is on and light is off. We found that when we turned on these somatostatin chortle cells, we increased delta power, a measure of cortical synchrony, across all layers of the cortex. We can do this regardless of the state of the animal. We can deepen the level of synchrony in both non-REM and quiet wake states, and also increase synchrony even in periods when these somatostatin chortle cells aren't typically on, such as movement. We looked at a number of other measures of the synchronization state of the cortex. We looked at the average pairwise spiking synchrony, the average cross-parallelogram of all the units we recorded from, found that neurons were firing more synchronously with one another during optogenetic stimulation. We looked at how these spikes corresponded to the ongoing local field potential fluctuations and found that they were more likely to fire in phase with this synchronized state during stimulation. And finally, we looked at down states, which are a feature of sleeping brain activity which are characterized by this positive deflection in the local field potential at the same time as a pause in firing activity. And when we stimulate these somatostatin chortle cells, these down states become more intense. So altogether, this is painting a picture where when we turn on these cells, we're making this local area of the brain seem more like it's asleep. We're increasing the synchronization state really, really locally around our stimulation. And given that we can see this, we started to ask the question, what happens to the rest of the animal? The animal is still in whatever sleep or wake state it is here, but if we activate these cells across the entire brain, can we start to change the behavioral state of the animal as a whole? And in order to ask and answer this question, Joffrey and I came up with this somewhat crazy experiment where Joffrey would go in and a teammate in the lab would go in and inject in 22 different sites in the mouse brain in order to express this excitatory dread receptor across all the somatostatin chortle cells we could get our hands on in this brain while sparing any subcortical circuits. And what this dread receptor does is when we inject a drug into this mouse, it'll turn on all the cells that we targeted with this tool. So we first started by validating this tool, and Joffrey put this mouse that's expressing dread receptors in the somatostatin chortle cells on the 2-photon microscope, injected it with this drug, and found that even in these periods of high arousal with really, really dilated pupil, the somatostatin chortle cells are really, really still quite active, suggesting that we're able to activate them. And we next wanted to ask what happens to state as we activate these cells across the entire brain. So we we put them in an arena, we injected them with either a vehicle, a non-drug control, monitored the sleep and wake state of the animal in this experiment, and compared it with a drug trial where these somatostatin chortle cells are activated. And when we activate these cells, we found very interestingly that animals slept a lot more. They spent much more time in this non-REM sleep state at the expense of time spent in wake. and also fell asleep somewhat more quickly after this injection when activating these somatostatin choroidal cells. We wanted to see whether or not this was some sort of physiological sleep, whether these animals were going to sleep naturally, or whether maybe they were just knocking out on the side of their cage. So we monitored them shortly after injection with videography. This is the same mouse you're seeing on subsequent days. And after injection of our CNO drug, where we're activating our somatostatin choroidal cells, this mouse is engaging in really different behavior. It's building its nest here and preparing to fall asleep. And shortly, it should stop moving, and we'll know that it's asleep. Whereas in the same period, just one day before, this mouse is moving around, still really active and not sleeping during this time. All this is suggesting that we're engaging natural circuits for the generation and promotion of sleep. And with that, I'll summarize what I've told you from this project. We've studied these somatostatin choroidal cells, this really unique population of inhibitory neurons. These cells project widely across the neocortex, targeting many downstream areas. During alert states, these somatostatin choroidal cells are largely inactive, and cortical networks are largely desynchronized, as the somatostatin choroidal cells are not able to act on local cortical circuits. However, during more quiet states, these somatostatin choroidal cells are really quite activated, and cortical networks are synchronized, as these cells are able to act on these local circuits. We used optogenetics to demonstrate the causal role of these cells in generating this synchronized state, and we're able to break the relationship between the synchronization state of the cortex and the more broadly generated arousal state, suggesting that these cells act as a sensor of this state to affect local cortical activity to reflect it. But the arrow doesn't only go in this direction. We also showed that the activity of these somatostatin choroidal cells feeds back onto the behavioral state of the animal and challenges this model of this subcortical only generation of sleep, suggesting that the cortex is able to play a more active role in shaping sleep and wake states. And that's all from me. I'll thank the lab and collaborators, all the people who made this possible, and then also take your questions and reserve some more acknowledgments for after.
All right, so the floor is open for discussion. Let's see.
You didn't know what you were getting into today.
Wow, that was such a great presentation, Jacob. My question was, maybe I guess I'm asking for a little bit of speculation if you have it, but you mentioned that there's like different phases of sleep, and although you showed really compelling evidence that these somatostatin choroidal cells promote sleep, do you have any idea of what phase of sleep they may kind of initiate, or do they affect do they change the proportion of different phases of sleep?
Yeah, it's a good question. You might have been a reviewer on our paper. Yeah, so they're active during the non-REM slow wave sleep phase of sleep, but we're looking into, and we were asked in our revisions whether they're maybe promoting longer bouts of sleep, whether they're more active during particular periods, the beginning of a bout of non-REM sleep or the end of a bout of non-REM sleep. They correlate strongly with the synchrony that we observe during sleep, so it's more likely that they're activated during these more synchronous phases of non-REM sleep, potentially maybe the earlier periods, but we haven't looked in detail at that yet.
Thank you.
Questions?
I agree with Aubrey, really nice presentation. Do you know who's talking to those SSD choroidal cells?
Yeah, it's a really good question. It's one that we've had for a long time. We've tried to do some rabies tracing, which will allow us to identify the upstream cells. We were expecting to see maybe something like I showed in that review article, some of these classic sleep generating circuits. We didn't see any of them in the rabies tracing experiment. What we saw was local excitatory neurons, really local to the area in which we injected the rabies tracer. Some cells in visual thalamus, and that's it. So this doesn't mean that there's not some connection from these sleep generating circuits, but we don't observe it in rabies tracing. We've got some sort of candidates that we've been going through in some slice experiments, but now that Stefano's left us, these have slowed down a little bit.
And I have one more, just a kind of a broader, more philosophical. So, and I think it starts from, benzodiazepines promote sleep, right? That's one of the things they do. So I'm really perplexed at that Allen Brain Atlas that the majority of SST are correlated with increased activity.
Mm-hmm.
That seems, I guess, but I don't understand the field very well. So let me ask one thing. If you had, say, targeted in the large scale manner that you did any other inhibitory cell and you just turned them all on, would you also get sleep?
Yeah, it's a really good question. And other people have done really similar experiments to this, actually. And an interesting one for interpreting our findings is when you do this with PV cells. So PV cells, powerful source of inhibition in the cortex. When you turn them all on, the animals do in fact sleep more, similar to the effect that you see with benzodiazepines, but the synchrony is actually reduced during this sleep. So they're sleeping more, but it's a different quality of sleep. They're not sleeping with the same intensity, I guess you might say.
Cool. Thank you.
First of all, amazing talk, and it's amazing to see how much work you've done. I was really interested in, like you said, the disconnect between the classical view of subcortical control of brain state and these wonderful cortical cells. So I was wondering just if you have a hypothesis of how those two modes of control interact with each other. Are they concurrent? Are these cells able to understand something about the cortex and then relay it to the subcortical areas? I know you only saw a small projection. Yeah.
Yeah, that's a good question. It's a lot of stuff in there. So we're definitely not the only ones who are interested in this cortical control of sleep. In the past few years, there have been a number of publications out there showing that the neocortex plays a variety of roles in this top-down control of sleep, which I think at a sort of intuitive level, I think makes sense. Maybe if you were out here in the audience and you didn't know what was going on during this talk, you might have been a little bored, and something in your cortex is potentially promoting you to be a little bit sleepier. How exactly these interact, I don't think we know. It could be through a variety of different ways. There could be direct projections from these somatostatin choroidal cells in a different area to a sleep promoting circuit. It could be through multiple interacting pathways, and sleep is really broadly distributed state in the brain. I think there's a lot of interesting work in the future that will go into disentangling exactly this kind of question.
So in sleep, there's supposed to be a replay of cortical activity and dreams, right? Do you think that your findings are related also to inducing sleep, but what happens during sleep?
Yeah, it's a really interesting question. So this replay during dreams is classically associated with the hippocampus and sharp wave ripples. There's a really similar population of cells in the hippocampus that are active specifically during ripples. They're genetically fairly similar, long-range projections, the whole thing. we have some data where we've recorded ripples at the same time as these, and somebody at some point will probably take a look at it and see if there's some sort of coordination between the hippocampus and the cortex where these cells are helping with these replay phenomena, but we don't really know right now. Perhaps that's what the subiculum projections are for.
So anatomically, you've well characterized what a long-range projection is. But what about physiologically? So if you measure, say, an LFP and you stimulate optically in one small area, how far away do you see synchronization?
Yeah, a lot of paper reviewer questions today. So we don't know is the short answer. We've been doing some experiments to try and figure this out. I did some experiments where we implanted these multi-shank electrodes, which have a stimulating fiber on one shank and then shanks that are spaced about 1 millimeter apart. In doing these experiments, we don't see any like drop off of the effect across this 1 millimeter of stimulation. So it at least goes that far. And now Joffrey and I have developed a prep where we're trying to record across longer distances to see if we can see where exactly this drop off occurs.
Do you have a sense if these cells have a different activity depending if the animal is sleep deprived or not? Because you're talking about like quality of sleep, and I'm thinking if they're tuned on just when the animal is super deprived or not, or something like this.
Yeah, it's a great question. So some of the previous work that I showed can speak to this a little bit. The work from Tom, who I think is here, he's shown that the amount of phos that these cells express is definitely correlated to the amount of sleep need or how sleep deprived the animal is. We've done some preliminary experiments where we've recorded from these cells after a long period of sleep deprivation, and it seems like the only difference that we can see is they're just more active. They're brighter in our recordings. But there might be some more other more subtle differences in their activity other than just being more strongly activated based on sleep need.
But I mean, I think it's related to the other question, like the ratio of phases of sleep might change maybe if the animals are sleep deprived, right?
Yeah. So there's definitely more slow wave sleep, more synchrony when the animal are sleep deprived, which would make a lot of sense for these cells being more active at the same time.
Thank you.
Hey, nice talk. Do these cells have other properties that you find in other hypothalamic systems that actually that have been known to generate sleep, like ADP buildup, sensors for that, or some kind of circadian rhythm present within these cells, so they could be themselves pattern generators instead of having to receive input?
Mm. So yeah, these cells, taking this sort of one piece at a time, they like all cells in our body, they express circadian genes. But there's this classical work that I didn't really go into today, suggesting that there's both a circadian regulation of sleep as well as a homeostatic regulation of sleep. We believe that these cells are more important for this homeostatic part, whereas the circadian part lives in like the suprachiasmatic nucleus and these gene expression cycles that we know about. Do they exist in other areas? I think was part of the other question, which there are, as far as I know, these cells have not been found in like these classic sleep generating circuits, like in the hypothalamus. But
The mechanisms from hypothalamus sleep generators, are they?
That's right. So yeah,
These
there's
things,
there's some work suggesting that adenosine buildup in like the basal forebrain is important for this homeostatic regulation of sleep, which is interesting. We thought about it for these cells in our rabies tracing experiment. We actually do see astrocytes, which are are potentially something that releases adenosine, but we don't know yet. We haven't looked.
Did I get the last question? So, wonderful, Jacob. That your work has inspired many, many questions in my brain, and I thank you for that. You took us in this beautiful evolutionary path, and I was wondering if you could speculate a little bit on the function. I assume all of these animals sleep, but you alluded to it yourself, like PV cells, it's about the synchrony. So what would happen if you ablate the function of these cells? Do you think it'll have like a cognitive phenomenon on learning, or sleep itself will be disrupted, or what's the pressure there?
Yeah, it's a good question. What's the necessity of these cells? Why are they kept around? I think the best thing out there to answer this question. There's these experiments where they did a whole body knockout of nNOS. So these cells are one of the major sources of nitric oxide in the neocortex, and they found that these animals were no longer able to respond to sleep deprivation appropriately. They didn't have this more intense sleep after sleep deprivation. So potentially what would happen if you totally lost these cells is the animals wouldn't be able to respond to this challenge as well and recover quite as quickly from being sleep deprived, potentially influencing memory factors and many of the other things that happen during sleep recovery sleep.
Thank you, Jacob. Circling back to the input question, in the absence of any rabies detectable inputs on these cells, do you know if there are receptors for neurohormones or other circulatory modulators that might be stimulating the action?
Yeah, it's a good question. We've gone into the single cell sequencing data sets and looked for these. There are some things that we might expect. There's muscarinic acetylcholine receptors that are probably involved in turning these cells off during high arousal states. Looking for sleep receptors for sleep promoting substances has been a little bit more challenging. There's a lot of sort of missing data points in these G protein couple receptors in the single cell sequencing. So we don't have a lot of great targets to go after there. But we'd love it if we did. But yeah, we'll get there.
Thank you.
So no more questions? All right, you may proceed with the rest.
Okay, so yeah, I'd like to take a moment here to thank all the people who helped me and supported me and brought me along in this really like intense special journey that was doing this PhD. I'll start with talking about Renata. Thank you so much, Renata, for taking me into your lab, for teaching me so much, for giving me all the support, anything I could have asked for to be successful, to be the best scientist I could be in the moment. I think it's a really special thing that I feel that you've given me, and I really appreciated our time together. It felt sometimes like you knew what to say to me before I knew what I was even thinking, and I think it's a really special thing that I found, and I feel lucky to have joined your lab. So thank you. I'd also like to thank all the other people in the lab. As Renata showed us at the beginning, like we've been a really tight group. I've had a lot of fun in these years with all of you guys, and I really appreciate all the times we've had together, and I'll keep them with me for a really long time. lots of other friends I've made along the way, and friends who have been with me for a while. Thank you all of you. There have been so many special moments, and I thank you for being there with me when things were tough, when things were good. It's been a lot of fun. A special shout out to the trash group. You guys were like the home base for me during this. Like, there's so many fun memories during this time that I shared with you. And I really hope we get to continue making these fun memories. Please come visit me in London. And I hope to return here to visit you guys as well. Thank you to my family, my New York family, and other ones. It was really a great experience to get to live in the same city as some of you, to see you a lot more often. I really enjoyed all the times we got to spend together. Grandma, I hope you're there on Zoom. I hope you enjoyed seeing this, and thank you for supporting me during this. I want to say thank you to my partner Kiefer. He's not able to be here today, but I really had a lot of fun with you these past few years, and I thank you for dealing with me when I'm going through these stressful moments that the PhD presents us with, and I look forward to making a life together with you in these years to come. And together we have our daughter that we're raising together. So thank you, Kitty. And then finally, I'll say thank you to my parents. You guys gave me the base to be able to be my best self in this life. I grew up in a place that really valued this curiosity, and it served me really well. Thank you guys. Thank you all for being here.